2 in 3 Aussies will be diagnosed with skin cancer during their lifetimes, making the sun one of our biggest threats. But why can a little bit of sunlight hurt us so much? Well, it turns out that UV rays actually harm anything with genetic information, including these guys. Meet Eric the E. coli and Tim the T1 bacteriophage. Eric here is an Escherichia coli cell, while Tim is a type of virus that infects E. coli. As you can imagine, these two aren't the best of friends, but one thing they have in common is possessing genetic information, specifically in the form of deoxyribonucleic acid, DNA. DNA is like a language of 4 letters, each corresponding to a different DNA subunit. 2 strands link up together to form the famous double helix shape, with A pairing to T and G pairing to C. But sometimes sunlight can get in the way. As discovered in 1960, UV rays cause the formation of pyrimidine dimers, a mutation in the DNA where C's and T's pair with each other incorrectly. Luckily, they can be fixed via a process known as photoreactivation. Photo refers to the use of light, meaning that, ironically enough, some organisms like Eric can use the very same UV light that causes these mutations to later repair them. It involves an enzyme called photolyase, which cuts the dimer linkages using light energy. But what happens if there's no light available? As it turns out, some E. coli strains have another mysterious way of fixing pyrimidine dimers. Remember Tim? Well, when E. coli cells are infected with T1 bacteriophage, they usually form plaques on plates of growth. These are areas where the bacteriophage has successfully killed E. coli cells and stopped their population of that area. When this T1 bacteriophage has been exposed to UV light, you would expect its DNA to be damaged, therefore reducing its ability to fight and conquer the E. coli cells. But work in the early 60s showed that somehow, the wild-type E. coli strains, the ones that could repair their DNA in the dark, actually showed the presence of many plaques, but not every strain had this ability, and in fact, less plaques formed on these strains. Following this, Paul Howard-Flanders, Richard Boyce, and Lee Theriot figured that these results could be explained if the UV-irradiated phage DNA is reactivated in wild-type E. coli, but not in these UV-sensitive strains. While this might not be the best strategy in a fight, it's certainly an interesting test of what genes control this new light-independent repair method. And so using this, these scientists set off to work out exactly what was going on with this mysterious repair process. First, they accurately mapped the 3 known genes that controlled this process: UVRA, UVRB, and UVRC. Using interrupted mating tests with 23 different strains of E. coli, they were able to produce the following genetic map. Next, they set off to gather additional data on plaque formation, as well as directly testing excision of thymine dimers in the bacterial cells. To measure dimer removal, the bacteria were grown overnight at 37 degrees Celsius in radioactive H3-thymidine. Samples were diluted to a 5 ml cell suspension, then irradiated with UV using a 15-watt low-pressure mercury lamp. After growth, samples were analysed using descending paper chromatography and passed through a strip scanner to measure the distribution of thymine radioactivity. The results showed no thymine dimer excision in 17 out of 23 strains, which included the 10 UVR mutants that formed the smallest number of plaques with the UV-irradiated T1 bacteriophage. They found that a mutation at any of the 3 genes causes a loss of ability to excise thymine dimers, making it unlikely that different kinds of dimers are excised by different enzymes. The results for majority of strains suggested the existence of an excision endonuclease, an enzyme able to excise mutations from phage or host DNA, which is coded for by the 3 UVR genes. But the functions of the products from the 3 genes remained unknown, and they didn't detect any sequential action at the time. Although this meant that they alone had not yet solved the puzzle, the 1960s were an exciting period of discovery. At the time, cutting and replacing sections of the DNA backbone was unheard of, but papers such as this led to the discovery of nucleotide excision repair, or NER. This resulted in Howard-Flanders and Boyce publishing a paper in 1966 describing the 4 known steps involved in NER in E. coli. We now know that the 3 genes are responsible for coding 3 proteins involved in NER. But how does it work? In E. coli, it starts with a protein made of 2 UVRA polypeptides and 1 UVRB polypeptide, which recognize and bind to the mutation, using energy from ATP to bend the DNA. The 2 UVRA proteins are released, and a UVRC protein binds to the UVRB DNA complex to cut the DNA on either side. Finally, DNA helicase 2 releases the excised nucleotides, DNA polymerase 1 fills the gap, and DNA ligase seals the molecule back together. And with that, the mystery method of DNA repair is solved. Just like Eric, us humans are capable of NER. but our pathway is much more complex. Still, we shouldn't rely too much on the wonders of genetic repair, so always remember to slap on some sunscreen next time you visit the beach.